Hi, so I'm Tony Ricci, and I guess we're going to have a little change of pace now, back to more of a basic science type talk. I'm really going to give kind of a broad overview of what we've done over the last year or so, what we're kind of in the midst of, and hopefully a bit of where we're going. I'm just picking out kind of tidbits here and there of the actual data that we're working on, just to try to give you a feel for what it's like in the lab. So, over the last year, we've had a major turnover of people in the lab. Bifeng has left. He became very Americanized while he was here. Jihan has also moved on, as has Manolo Castellano, moved back to Spain. And Marcus Huth has also gone back to Europe. Marcus came into the lab really quite handicapped. He was the first German to enter our laboratory. And he was also our first clinician surgeon coming in to attempt some basic research. So despite these handicaps, he actually did quite well, published several papers, and it's a big loss to have him go. But we also have new people moving in, and so recently Connie has joined the lab. Connie also, in the short time she's been there, has several distinctions. She's the first neuroscience graduate student to be in our lab. She's also the shortest member to ever be in our lab. This comes in very handy because she fits in places no one else can really get to. She's also somewhat of a chameleon, being able to change hair color almost on a regular basis. So you can almost sense her mood based on the color of her hair. Mamiko has also joined the lab recently. Mamiko is just slightly taller than Connie. It was a double-blinded study to figure that out. I have every confidence of Mamiko's success because she's a baseball fan, which is obviously important to me, and an American League fan, which is even more important. So that's good. And Eduardo has also recently joined the lab. So all of you that know Eduardo can feel my pain here. But some things also are staying the same. So Mike has been in the lab now for he's been in the lab for about a dozen years. This was Mike's reaction to hearing that Eduardo was joining the lab. We also have Anton. Anton has been with me for about 3 years, but he was with Stefan for, I don't know, a lot of time, 4 or 5 years before that. So he was actually one of the founding members of the research division because he came at the same time as Stefan did. We also have Felipe. Felipe's kind of an in-between because he's running the imaging core. He's also working with a variety of us on different projects, and you'll hear from him later. And we also have Thomas, who's our second German to be in the lab. He insisted that I show his image with meat, and I think that kind of speaks for itself. Okay. So, and of course, no lab is successful without the constant harassment of the administrative staff. I mean dedication and great knowledge base and patience with all of us. I'm just going to kind of do a little overview. So, of course, where we are with funding always is important. So we just renewed our transduction grant, so that was a relief. And this year we are trying to renew our synaptic grant, and we're debating whether we're going to try to turn our R21 aminoglycosides into an R1 as well. We've also had some funding from internally through the SPARK program and also BioX. But I want to also pay tribute to the other people in the lab who've also working hard toward getting funding. So Anton has had an NRSA for the last couple of years. He's also applied for a K99 this year, which is kind of the natural progression. Thomas has a grant from Germany. He's also for the last year, and he's also renewed that, and he's applied for internal funding, and we're about to apply for another German funding. And Connie also has a NSF graduate fellowship. Mamiko has applied for an internal grant, and she's also applying for an NRSA this fall. So really, it's kind of a team effort. Everyone's kind of trying to pull their weight. There's also work that's kind of joint between grants, and you'll see as we go through that there's a lot of kind of interactions between labs. So that I'm kind of a co-PI by default on a grant with Sunil. I also help out with on a couple of Stefan's, and I'm direct the imaging core on the training grant. All right. So these are the publications we've had over the last about 18 months or so. I'm not going to go through all of them, but you can see. So there's some on transduction, there's some on synaptic, and there's a couple on aminoglycosides. So we're really kind of have our hands in a variety of different pots. These are the papers that are either in revision. So we have 6 papers that are kind of directly in the process, either in revision or have just been submitted. And we have 3 papers that should be hopefully submitted before the end of the year. 2 of them, I think, are really kind of paradigm shifting type of papers, and I'll talk a little bit about each of these as we go forward. So there's really 3 directions in the lab. This is our synaptic transmission cohort of people. Felipe also works on the synapse with us as well. This is our mechanotransduction group of people. And then we have some translational work, and mostly at this point it's aminoglycosides. And this is really, it's hard to separate, to say, well, this is what we do and this is what Alan does. It's really just one big group effort. So there's Alan with his stern look. And then Bob Greenhouse, who's our chemist. And so this is really the aminoglycoside side. And then we have Eduardo, who's also in the lab. And we have Nick Blevins, and we're working on kind of some endoscopy work with new ways of imaging the cochlea. And Eduardo's going to go into more detail on that, so I won't. So first I'll talk a minute about synaptic transmission. So the ear is really remarkable in its ability to communicate with the brain, because the synapses, which is the way the hair cell connects up to the brain, can operate at very high frequencies for long periods of time without fatigue. And it can do this while maintaining very precise timing information. Now, how this happens is really a mystery at this point. We know more that it happens than actually the molecular mechanisms of it, and this is one of the areas that we're interested in studying. We do know they have this, what's called a ribbon synapse, as do many other sensory cells that work with a graded type of receptor potential. And we've spent a lot of time trying to unravel the molecular mechanisms of how this works. And the research has taken a little bit of a change in that we're really now going to start looking more at what's happening postsynaptically, a little bit more at the systems, which is, and so the basic question is kind of what is responsible for dynamic range. And the way to think about dynamic range is that, you can hear a pin drop and you can hear a lawnmower, and neither one of them is really doing you any damage or saturating the system. And there are some fibers that allow you that have this low threshold, and there's some fibers that have this higher threshold. And how this happens, we don't know. And so over the next year or so, we plan on figuring out how that happens. Or more precisely, Connie and Mamiko are going to figure out how this works. We're also interested in what underlies the temporal fidelity of the system. So you can think of this as phase locking, if you want. And Mike has spent a lot of time now developing the ability to record both from the hair cell and the nerve fiber simultaneously, so that you can study directly the input-output functions. And so we're going to put a lot of effort into trying to understand first at the functional level, and then hopefully at the molecular level how this works. And an ongoing project has been understanding actually how vesicles are moved around presynaptically, and what exactly does the ribbon do, and what are the proteins that are specifically involved, and what are their specific functions in the process. And so we have done a bunch of the physiology, and now we're trying to incorporate more of the molecular component to that. And also, we've been looking at synapse maturation and maintenance, and this is important kind of across labs. And so this is really more of a group effort approach. But I'm going to take a minute now just to talk about some recent data that's submitted that Mike has. And so this is recordings from a nerve ending in a turtle, auditory papilla. And what you're looking at are called EPSCs, which is basically release from the hair cell onto the nerve. And you can see they come in different sizes and different shapes. And one thing to know, or important difference between the turtle and the mammal, is that in the turtle you have one fiber with multiple synapses, whereas in the mammal you have multiple fibers, each having one synapse. And it's really not understood what the benefit or what the difference is, or why did you go from one to another. And so what I'm going to show you in a minute here is simply an idea for what the benefit is, and why, in fact, the mammal needs this one synapse per fiber, and why the turtle can actually work in this lower frequency range with these multiple synapses. So if you break this down more, what you find is that you have these single events where there's just a one rise time and one decay time. And these can come in a variety of amplitudes. But you also have these, what we call complex events, where you can see there's like a hitch in the response, which looks like there's maybe two vesicles that are timed close together. Or here you get three events where nothing comes back to the baseline. And here you have multiple events. And you can see that when you have multiple events, the total gets bigger. Okay? And what we think is happening here is that this is all spontaneous activity. So what we think is happening is that these different synapses are all releasing roughly close to the same time, and when they do, you get this summation. And so if you look under more physiological conditions, meaning that you measure action potentials and the membrane voltage changes, then we can basically try to look at which of these types of responses are better at generating the action potential, which is what's going to the brain. And so firstly, you can see that about 40% of the events generate an action potential total. Of the action potentials that are generated, more than 70% come from this complex grouping of action potentials. Okay? And if you look at the complex events, it's almost 90% of them generate an action potential. So they're highly efficient at generating action potentials when you group them together, compared to when you get these single events, even though you can get a variety of amplitudes in the single events. And so what we think happens, and I'm not going to go into too much detail, is when you stimulate this fiber or the cell, you basically you synchronize the timing between these different synapses. And so you selectively get this type of summation, which brings you across threshold and generates action potentials. So the benefit of that is you get this timing, you get these action potentials. But the problem with it is you get this big summation effect, which takes a long time to happen. And so it's going to limit how fast you can actually go and what type of timing you can get. And so the turtle can work up to about 800 or 900 hertz. But when you get to the mammal, where you have to be able to work in the kilohertz, or even in the bird, where you have to get up to 5 or 6 kilohertz, you lose the number of synapses, and you need to get timing within one given synapse as compared to across synapses. And so we're now using both the mammal and the turtle to try to compare the specific release mechanisms there to understand how you get better timing in the mammalian synapse compared to the turtle. All right, I don't want to take too much time. So there's also collaborative projects on the synapse. So we've been working with Joe. Everyone knows Joe from the Christmas party. That's when he typically comes out to visit. We're working on technology for measuring single vesicle release. It's a project with Stefan and Micah looking at CAM kinase and otoferlin. And there's posters for all of these, so you should go look at the posters. We're working with Mirna to look at the role of thyroid hormone in synapse maturation. And then we have Jamie Adelson in the lab. She's kind of a Klingon at this point, who's using some of our toys to look at plasticity in the visual cortex. All right, so then transduction side. So transduction is how you convert a mechanical signal into an electrical signal. And so if you deflect the bundle, you pull on these links, and it opens channels. And you generate this current, and this current has this decay, which is called adaptation. Okay, and we've been studying that for years. So I'm not going to list this, but there's a variety of projects that are ongoing in the lab, trying to understand the mechanisms of adaptation, the role of the lipid bilayer in lipid, trying to figure out what the actual channel is that's in there, and how the channel is activated. But I'm going to show you one kind of startling piece of data that's also under review at this point, and that is that in the mammalian cochlea, calcium does not affect adaptation. So since the mid-'80s, it's been largely accepted that calcium entry through the transduction channel is responsible for adaptation. And this is either by directly interacting with the channel to cause the channel to close, or by binding it to a myosin, then myosin drags the channel up and down the stereocilia. And so the paper that we have in review right now basically says that neither of these are valid. So this is adaptation at negative potentials. When you go to a positive potential, that means calcium cannot go in the cell. And when calcium can't go in the cell, here you still see adaptation. And if you do the classic experiment, which is just do an activation curve, get adaptation, and then do another activation curve, you shift. So this is how you extend the dynamic range. But if you do this experiment at a positive potential, you get exactly the same thing. So there's a bunch of other data that goes with this, but it basically says that calcium cannot be regulating adaptation in the mammalian cochlea. So I think it's kind of the end of an era when it comes to myosins climbing and slipping. So there's a lot of people involved in the transduction project. Peter and Uli. This is Robert when he heard that Eduardo was joining our lab. A variety of other people. And there are a lot of projects going on. And if anyone's interested, they should just come and talk. And there's posters on a bunch of these. And then I'm just going to do a quick one. So our translational side is one looking at aminoglycosides and also developing some technology. And so the technology part Eduardo is going to talk about later. And so what we spent a lot of time on is showing that aminoglycosides, hair cells are sensitive to aminoglycosides because they go through the mechanically gated channel. And so we've pretty much established that, I think, as much as it can be established. And so we've been studying this channel for a long period of time, and so we have insights into it. And so we designed some new aminoglycosides that theoretically shouldn't go through the channel and so should prevent ototoxicity. And in our first round, I'm just going to show you an example of one of these compounds. We have a whole series of these compounds. So this is a control tissue in culture. You can see 3 rows of outers and a row of inners. When you put kind of the parent aminoglycoside on, all the hair cells are dead. When you put our new compound on, all the hair cells are alive. So if you count the number of hair cells in 3 regions, this is control, so they're all there. This is with the parent compound, and so you see they die. They die in a base to apex gradient. That's all been known. When you use our compound, they all live. If we do a dose-response curve, this is the parent compound. If you do a dose-response curve with our new drug, if you go up to about 3 millimolar, you can start to see some damage. But in general, there's more than a 10-fold change in toxicity with no effect on antimicrobial activity. So it kills bugs just as well, but it doesn't kill hair cells anymore. So that's just kind of it in a nutshell. I think it may or may not take a village, but it certainly takes a team effort to be productive and successful. And I'm just happy to be part of this team. So thanks.